Just like you and I have a multitude of ways to communicate, phone calls, texting, emails, face-to-face, cells as well have many ways to communicate. We call this signal transduction. In this lesson, we will meet the following objectives. First, we will define signal transduction. Then we will look at the structure and function of G protein coupled receptors, gated ion channels, and receptor tyrosine kinases. Lastly, we will take a look at how these receptors and channels function in the nervous system, cancer, and in regulation of the cell cycle. Before we jump into the specifics, let's stop and take a look at what signal transduction is. It's how cells take information and turn it into chemical change or cause a chemical reaction. These signals cause a cellular change. Signal transduction is specific to each type of cell. The process is also sensitive. Because the molecules that bind to receptors, such as ligands, have high affinity for receptors, this participates in the specificity and sensitivity. And then this cooperation between the molecules and the receptors causes large changes. It doesn't take much, very small concentrations sometimes of ligands to affect the receptors and cause significant changes. Now, once this receptor or enzyme is activated by a molecule, it triggers the activation of another enzyme, which affects another enzyme, and so on. This is known as an enzyme cascade and greatly amplifies the message, thus amplification. Signal transduction occurs through 4 types of receptors: G protein coupled receptors, ligand-gated ion channels, and enzyme-linked receptors in the cellular membrane. We also find nuclear receptors in the nucleus. Let's start with an examination of the G protein coupled receptor. In order to understand how it works, you need to understand its structure first. So what we see in this diagram is that it is a 7-transmembrane receptor. That simply means it spans the membrane 7 times. The amine terminal is outside of the cell, and the carboxyl terminal is on the inside of the cell. Now, this receptor works with something called GS, or stimulatory G protein. It has 3 parts: a beta, a gamma, and an alpha subunit. This is key to understanding how the signaling process works. Sometimes we can get inundated with the details, so it's nice to step back, and look at the big picture. There are 3 main components to signal transduction and a G protein coupled receptor. First, you have the receptor. Now, this is that 7-transmembrane receptor, and it's made up of helical segments. When this ligand binds to the receptor, it then interacts with a G protein. Now, that G protein, part of that G protein, it has 3 subunits. Part of it's going to come off, and it's going to go and interact with a different enzyme or an ion channel, which is going to then continue the signal on down the line. Let's take a more specific look at how the G protein coupled receptor works. First, there is a signaling molecule that binds to this 7-transmembrane protein. When that happens, it causes the GDP that's on the alpha subunit to become GTP. And when that happens, the alpha subunit becomes activated. It dissociates from the rest of the GS protein, and it can go on to trigger another molecule. In this case, adenylyl cyclase, that is hard to say, which then activates ATP to make cyclic AMP. Cyclic AMP goes on to activate a protein kinase, which goes on to phosphorylate something in the cell. So this is a really great example of kind of a cascade that we see in the signaling. A molecule starts right here, which triggers this, which triggers this, which triggers this, and so on and so forth. Luckily, it's kind of like that game telephone, but typically the signal doesn't get as lost as it does with a bunch of 6-year-olds. So we've already looked at the G protein coupled receptor. Now let's take a look at the gated ion channels. But first, let's step back and look at the big picture. Things need to get across the cell membrane, both into the cell and out of the cell. But how do they do that? Well, ions have kind of a tricky situation. They're charged, they're polar. The inside of the cell membrane are made up of lipids. It's nonpolar. So the ions can't just go diffuse right through that part of the cell membrane. So they need a method to get from the outside of the cell to the inside of the cell. And this is where gated ion channels come in. Now, these are not transporters, but they're just channels with gates that open and close, just like a gate on your fence can open and close to let your dogs in or out. Now, there are 4 types of gated ion channels. There are voltage gated, ligand gated, mechanically gated, and thermally gated. But we're going to focus on the first 2, voltage and ligand gated. So how do ligand-gated ion channels operate? Well, typically they are closed, but they have a spot where a ligand can bind, kind of like a lock and a key, or what we call an induced fit. When the ligand binds, the channel opens. Interestingly, the binding site is not usually near the opening of the channel. Now, once it opens, ions like potassium, sodium, chlorine, calcium can move through the channel. When they move through the channel, especially if they're moving into the cell, it changes the electrical properties of the cell. We say that an intracellular electrical signal occurs when the ions move in. Now, there's 2 kinds of ligand-gated channels. There are fast and there are slow channels, and they differ in their protein and their response time. Fast channels have the same protein receptor as the rest of the channel. Thus, the response time is fast, like milliseconds. For the fast channel, the messenger is a ligand which binds to the receptor, which is the channel. The channel opens, ions move in or out of the cell very quickly. The slow gated channel, though, has a receptor that's a different protein than the rest of the channel. And this just results in a slower response, which could take minutes. The second kind of ion channel we're going to look at is the voltage-gated ion channel. Now, this is not to be confused with the ligand-binding ion channel, which works when a ligand binds with a receptor and the gate opens. The voltage gates open due to changes in the electrical potential across the cell membrane. And this can be controlled by differences in ion concentration on either side of the cell membrane, the difference in the electrical potential across the cell membrane, and believe it or not, ligands may have a role in this as well. This tightly regulated mechanism is controlled by the number of types and the number of open ion channels at any given time. Voltage-gated ion channels can create what we call an action potential. And the best way to see these in actions is to take a look at the nervous system. Nerves are a great example of voltage-gated ion channels because nerves carry electrical impulses, which are also known as action potentials, through their axons. Before we go any further, let's just take a quick review of the beautiful structure of nerve cells. They have a quite fascinating long structure, and depending where they are in the body, they can be quite long. Up at the top is the cell body with these dendrites that kind of stick out. And then the tail portion is what we call the axon. And then there is a space between this neuron and the next neuron. So we will call that space the synapse. Now, when the signal gets to the end of the cell, the neurotransmitter is released and it goes on to the next nerve cell and so on and so forth. So the way that these are lined up is quite important. Now, this picture here kind of takes a little bit of a deeper look at what's happening with this transduction signaling. So along the axon are voltage-gated ion channels. Now, normally the sodium channels are closed, but when the membrane is depolarized due to some sort of signaling molecule like acetylcholine, they open up. Right after they open up, the potassium ion channels also open up. Well, what's happening is kind of interesting. Sodium rushes into the nerve cell while potassium rushes out. Now, as that is happening and the signal gets lower and lower and lower into the axon, the calcium channel is then opened up. And you'll notice here that the calcium ions are flowing into the cell. That is actually what triggers the release of the neurotransmitter like acetylcholine into the synapse, which then gets passed on to the next cell. So the process begins all over again. So these three ion channels accomplish their task by changing concentrations of ions in the cytoplasm, and then these either act as second messengers or electrical potentials, which affect other membrane proteins on down the way. Another key piece in signal transduction is the receptor tyrosine kinase. This is an integral receptor protein. That means it's embedded in that cell membrane. Now, on the extracellular side, the outside of the cell, is the ligand binding domain. And you can see that right here in this picture. It's where this little red molecule has attached. On the cytoplasm side is the enzyme active site. Now, the ligand binding domain up here and this piece down here that's inside the cell are linked together by a single transmembrane helix. The cytoplasm side is what we call a protein kinase. And kinases' jobs are phosphorylation of residues. Or in other words, they're going to add phosphate groups from ATP to these residues. So when a signal molecule attaches, as you can see in this picture, the molecules become dimers. So what you don't see in this picture is that originally these two receptors are separate entities. But when those signaling molecules attach, these molecules join together to become a dimer. And this is what activates phosphorylation of the tyrosine regions. This sparks phosphorylation, which is the removal of a phosphate group from ATP and attaches to this region of the molecule. This is the first step in a signaling cascade. Now, when the dimer is fully phosphorylated, so all of those tyrosines have phosphate groups on them, relay proteins will come in and attach. And when they do that, those relay proteins undergo conformational change, which then allows them to release, and they go on and activate some other molecule to cause a response in the cell. And this is what will cause another signaling cascade. And this is roughly how receptor tyrosine kinases help cells communicate with each other. Receptor tyrosine kinases are vital in normal functions, including regulating cell growth, motility, differentiation, and regulating metabolism. However, through gain-of-function mutations, gene amplification, chromosomal rearrangements, and autocrine activation, these normally tightly regulated receptors are transformed, and there becomes a problem with the balance between cell growth and cell death. And quite often, this leads to cancer. Earlier, I mentioned that we were not only going to look at different types of receptors and ion channels, but we were also going to look at their role in nerve transmission, cancer, and finally the cell cycle. So receptor tyrosine kinases have a huge role in cancer. Let's take a look at that in a little bit more depth. Typically, cancer could be summed up as cell growth gone out of control, cell growth gone crazy. I know, sounds like a spring break slogan. But let's look at how four signaling ways can get messed up, which promotes out of control growth and cancer. Now, this first picture here represents gain of function. A gain of function mutation causes a normal product of a gene to be expressed inappropriately, usually at very, very high levels, or maybe even at the wrong time or the wrong location. So there's some sort of acquisition of a new abnormal function through some sort of gene alteration in itself. And this just messes up the whole downstream signaling mechanism. Many mutations can occur here, but one in particular is what we call small in-frame deletion, and that can make the kinases hyperactive, which then magnifies the downstream signaling. What's really interesting is sometimes the mutation can actually cause the receptor tyrosine kinase to become overactive without, look at that right there, without the binding of a ligand first. Kind of interesting. Another problem that could occur is what we call gene amplification. And this just refers to an increase in the number of copies of a gene in a genome. And this causes overexpression of receptor tyrosine kinases, or increased receptors, period. And then, of course, this increases the signaling. As you can see in this picture, we don't have just one, we have many receptors, and they're all activated. Every single one of them has a phosphorylated region. So this regulatory mechanism, it just cannot keep up. It just goes crazy, and it can cause out of control cell growth. Then we have the chromosomal rearrangement. And this can create brand new novel tyrosine kinase fusion oncoproteins. And this is actually what happens in the Philadelphia chromosome, where part of chromosome 9 is attached to chromosome 22. And this mutation is what leads to chronic myelogenous leukemia. This oncoprotein consists of two parts. So this kind of peachy color, maroon color, is the original receptor tyrosine kinase, but this aqua color here is its fusion partner. And this oncoprotein can be embedded in the membrane, as we see here, or just float in the cytoplasm. But either way, the chromosomal rearrangement can cause issues with signal transduction. Now, the last thing we mentioned in the last slide is autocrine activation. So normally what happens, growth factors and cytokines are released by secretory cells, and they go to some target cell. Well, autocrine is when the target cells secrete things themselves. They don't wait to be activated. They're activated on their own. And so these cells are no longer controlled and regulated and promote the tumors. As you can see in this picture here, there are many, 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 many more ligands than you would normally find that just keep repeatedly binding to the receptor tyrosine kinase, turning on the signaling mechanism. Because as you can see, you've got phosphorylation of the tyrosine regions. And if you remember from before, the relay protein can come in, bind to that phosphorylated residue, undergo conformational changes, release, and go on to create a signal somewhere else. So when these target cells can release these molecules on their own, they're unregulated, and we wind up with uncontrolled growth, which leads to cancer. So this takes us to, how is signal transduction important in the cell cycle? Now, when I teach freshman high school biology students, I want them to understand the big picture of the cell cycle. Basically, there's two parts. The cell is living and doing its job, and then it makes new cells. Living, doing its job, makes new cells. Now, it's a little bit more complicated than that. But here are the different parts of the cell cycle. You have G1, which is known as gap1, S for synthesis, G2 for gap2, M is for mitosis, and then sometimes there's a G0. So during G1, gap1, this is when the cell is brand new. It's growing, it's preparing for DNA synthesis, it's just doing its job. And then during synthesis, the DNA is actually replicated to ensure that there's enough DNA for two nuclei during mitosis. For example, in human body cells, there are 46 chromosomes. Well, if the body is going to make two brand new cells, there needs to be enough DNA to go around. So if I gave you 46 socks and I said, please make me two piles each having 46 socks. It would be an impossible task. But if you were able to make a copy of each of those socks first, well, then you can make one pile of 46 here and one pile of 46 there. And that's essentially what happens during DNA replication. Then during G2 or gap 2, the cell continues to grow. In fact, it about doubles in size, and it just does its job, whatever that job may be. As you know, all cells in the body have different jobs, and it's preparing for mitosis by making new proteins. Now, during mitosis, the genetic material is separated into two nuclei through a series of phases. In prophase, the nuclear membrane disappears. In metaphase, the chromosomes line up. In anaphase, the mitotic spindle or spindle fibers pull the chromosomes apart. And in telophase, the chromosomes go back into a nucleus, the nuclear membrane reappears. And then at the end of telophase, but in a separate process, is called cytokinesis. And that's where the cell actually splits and pinches off into two new cells. So you start with one daughter cell and end with two. Now, sometimes cells need to go into what's called G0, and it's kind of a pause. This pause may last for days, it may last for hours, it may last for the lifetime of the cell. It really depends on the situation, and that situation could be damaged DNA where the cell needs to repair itself, or it could simply be that this is how the cell spends its life. It just spends its lifetime in G0, never actually going through this whole process. Now, in the next slide, we'll take a look at how this cycle is regulated. The cell cycle is tightly regulated, and it's a pretty cool complex process. So I just want to focus on a little bit of the most important parts that are regulated. Now, first and foremost, the cell cycle is regulated through something called a CDK, and it is the cyclin-dependent protein kinase. But it doesn't act by itself. It has to act with this cyclin, and then together they combine and create a unit. But that's still not enough to make the cyclin-dependent kinase be active. So how are CDKs controlled? Well, first, there has to be a dephosphorylation and a phosphorylation of the actual cyclin-dependent kinase. We have to control the synthesis and degradation of the cyclins. And then lastly, there are some inhibitors that will stop CDK from doing its job. And I also want to touch on some of the other vitally important roles that cyclin-dependent kinases have in the cell cycle. So first, we need to understand what activates a cyclin-dependent kinase. So of course, you have to have a cyclin. And by the way, they're named cyclins because they cycle up and down throughout the cell cycle. They're made, they're degraded, they're made, they're degraded. When they combine together, you have a unit, a cyclin CDK complex or unit. Now, that is not activated, and here's why. In the cyclin-dependent kinase, there is a tyrosine 15 residue, and it's phosphorylated. It has a phosphate group, and that blocks the ATP binding site. Well, on the other side of the CDK is a threonine 160. And that gets phosphorylated. And when that gets phosphorylated, this tyrosine 15 dephosphorylates. So now that ATP binding site is open and ATP can come in and bind, and the whole complex is finally activated. Now, the cyclin synthesis and degradation is controlled through growth factors and transcription factors, and they are degraded by proteasomes. So as long as you can control how many cyclins are present, you can control how many get bonded to the CDKs. Now, there are lots of different CDK cyclin complexes throughout the cell cycle. We're going to focus mostly on the one that is in G1 or GAP1, but we also see cyclin E with CDK2 in synthesis. In GAP2, we have cyclin A in CDK2, and in metaphase, we have cyclin B in CDK1. Now, in GAP1, we have cyclin D and CDK4/6, and it's probably the most important piece of the cell cycle because it determines whether or not the cell goes through the division process. See, in G1, there's this point called the restriction point. And once the cell gets past the restriction point, it's got to go through with it. It's kind of like when you're having a baby and you're in labor and you're on the way to the hospital, you have to go through with it. There's no turning back. So how does this cyclin D CDK4/6 complex work? Well, it's a little bit more complicated than that. We have this protein called PRB, and PRB is normally bound to E2F. When E2F can dissociate, it can go bind to DNA. It can then transcribe a set of genes, and that will then allow replication to occur in the synthesis phase of the cell cycle. So if the cyclin D CDK4/6 is not available, it can't phosphorylate PRB, which will make it let go of E2F, which will then let it do its job. So without cyclin D, CDK4/6, the E2F can't do its job and synthesis cannot occur. Now, what if DNA is damaged in G1 and we don't want the damaged genes to get synthesized and made copies of, because then the new cells will have the mutation? Well, there is a protein that can then inhibit the CDK, and in this one it's called P53. Now, P53 activates P21, which then blocks the cyclin D CDK4/6. So synthesis will not occur because the PRB isn't going to unbind from E2F. And if E2F can't do its job, synthesis can't occur. Now, sometimes the cell will just go into G0 and stay paused for a little bit while the DNA gets replicated, while the DNA gets repaired and fixed so it can be replicated. Other times, P53 can cause apoptosis or cell death. Now, there's a few other things that CDKs are vital to in the cell cycle, and I just wanted to touch on them briefly. First of all, lamin is a component to the nuclear membrane. When lamin gets phosphorylated, it allows the nuclear membrane to degrade, and you know that is important in mitosis. The CDKs can also target actin and myosin, which are the contractile fibers that cause cytokinesis and make the one cell split into two cells. So without the cyclin CDK complex, cell cycle won't even get going. It won't get out of G1 if that very first complex can't do its job. And of course, the other ones have their jobs as well, but that's for a later video. As we have learned, cells have many different ways of communicating, which we call signal transduction. G-protein coupled receptors, gated ion channels, and receptor tyrosine kinases play key roles in that communication. Identifying and describing signal transduction's roles in the nervous system, cancer, and the regulation of the cell cycle shows even more so the importance of these receptors in the human body.